Welcome, class. Today I want to review with you mitosis. I'm doing something a little bit different today. Really I'm recording right on top of a website called Cells Alive. www.cellsalive.com is the site that I'm working from today. And it looks familiar because we also talked about it and you viewed this a couple of days ago in class. So it should be relatively familiar to you. But those of you who aren't, it's a great site to visit primarily because it has just about anything you'd possibly need to see in cells. This is meiosis, mitosis, talks about different kinds of cells. It's a great website, so you may want to go ahead and check that out. But today what I want to do is I want to really focus on mitosis. So I'm just going to click on the mitosis button, and then we have this animation right here. And I'm going to go ahead and play the animation. And as you can see, that it is an animal cell because of its, it just has a cell membrane, it doesn't have a cell wall. It's not like blocky. It's a little bit more of a roundish shape. It doesn't have a really defined shape. That's what's one of the things that separates an animal cell from a plant cell. So let's go ahead and watch it. You can see as it kind of runs through it, the main things that you need to watch for is the nucleus and the chromosomes. Those are the things you can watch that can pick up. A lot of other things are going on, but I'm going to concentrate primarily on what the chromosomes are doing and also what some of the other organelles are doing as well. So let's kind of talk about it just step by step. And we're going to start off with interphase. Now, before I do that, though, I just want to remind you that the entire purpose of mitosis is primarily to double or replicate its DNA so it can pass on to the next generation. So it's all about cell division. It has nothing to do with the generation of sex cells or gametes. It's really just replacing cells, like for instance when our skin sloughs off, or some other parts of our body just needs repair or replacement of certain cells. That's what mitosis is about. It just wants to replace that other cell or with more of the same kind of DNA. So it's all about dividing into two and making sure they're identical or exactly the same. So we're going to start with interphase, and I'm going to show you 7 steps. A lot of the books and it will show you 5, but I want to break it down to a little bit greater detail. And so we're going to start with interphase. Now, interphase is a little bit misleading because a lot of times they call it the resting phase. I really don't like to use that because it's far from resting. In this particular point, 98% of the cell cycle is used up in interphase. So that's a lot of resting if it really is resting, but it's not. It's preparing itself for cell division. So the things that are going on in interphase are like they're building proteins. They are kind of getting everything ready for that time when it has to split in two and have more organelles, so that when it does split, half the organelles will go to one and the other half goes to another. One of the things you don't see in interphase are the chromosomes. They're definitely still there. And to kind of give you an idea, I want to draw off to the side here. The chromosomes are basically the DNA, and it stretches in these long strands. So it's not coiled up or really condensed. And on each of these strands are, of course, the bases, and they have specific shapes depending on what is on there. And then the complement base will line up with this pair. These 3 things right here, and I'll talk more in detail on another video on the actual base pairs. This is called a codon. And that codon is being read by messenger RNA to make proteins. And as messenger RNA reads that, it gets an exact duplicate and makes proteins. But really, in cell division, we want another complementary strand of DNA on it. So it's gonna read the complementary DNA. Will read it across here, and actually will stay together with this codon. And then when it comes time to split, then the DNA from one cell will go to one side and vice versa. So this is kind of how interphase is. 98% of it stays in interphase, but at this particular point in time, chromosomes are doubling, or not quite doubling, but they are preparing themselves. So it can't be seen. So the chromosomes can't be seen in this one, but the cell is really doing a lot of other things going on. Now, in prophase, if you take a look at this phase, you'll notice that you begin to see these chromosomes. The chromosomes are beginning to condense. They're starting to roll up, kind of in coils, and they've already doubled. So in prophase, the DNA or the chromosomes have doubled and are starting to form in these kind of condensed, so we can begin to see them. And in this particular phase, it is doubled, so there's twice as much DNA in it. And the other thing that's going on in here, let me get that off of there. The other thing that's going on in there is that the nucleus is beginning to start breaking up. They have enzymes starting to break down all the proteins into amino acids. So that is prophase, and is primarily what's really going on in that particular phase is the chromosomes are doubling. Now the chromosomes, when they double like that, we have what we call tetrads, and those tetrads, they're kind of like this. They're all condensed down, they're doubled. And then right in the center, they're anchored by this centromere. So the centromere is going to be the place for spindle fibers. So it's kind of preparing itself for that. And so we have these tetrads, these like four, it looks like 4 pieces, but there's only basically, there's a DNA strand, there's another one, it's doubled. So that's called a tetrad. And of course, these are now the chromatids, and they're ready for dividing. But there's still got to be one more phase left to go, and before they actually split, and that is prometaphase. Prometaphase is that time where they begin to line up. The chromosomes begin to line up on the in the middle, or on the kind of the center plate here, just right here. They're beginning to line up, but they're not quite lined up yet. And of course, the centromeres are being attached by the spindle fibers. These spindle fibers are from the centromere, or excuse me, the centriole, and the centriole sends out the spindle fibers that attach to the centromere. So when that's happening, then we're getting ready for the next phase. And that next phase is metaphase. This one is really easy to see in a microscope because they're all lined up down that center plate. And in this particular case, you can see definitely the demarcation right down the center. And spindle fibers in place, the centromeres have been attached to them, and now we're ready for the next phase. And during this next phase, it's called anaphase. Anaphase is when the chromosomes are being pulled apart. The spindle fibers are starting to shrink down. As they shrink, it pulls the chromosomes to opposite poles. And of course, then you're beginning to also see right here, you can see this is starting to be pinched off. And you'll notice that, of course, the nucleus is completely gone. The nucleus got kind of completely dissolved, primarily back in prometaphase. Prometaphase and prophase is when the nucleus finally disappeared, and that was because inside the nucleus there were, and also outside the nucleus, there were other proteins called enzymes that start breaking it apart, and they're going to use it for other things. So that's anaphase, is when the chromosomes begin to break apart and move to the opposite poles. And then, when we get to telophase, that's the next phase, they have kind of reached their final destination. They've reached to the opposite end of the poles. Their spindle fibers have shrunk down to as much as they're going to get. Now, in this case, what happens is we begin to see 2 separate cells being formed. They're not quite split down the center yet, and they haven't shared all their cytoplasm yet, but they're getting to that point. Telophase is really about getting to the end of their travel. The chromosomes come to the end. And that brings us to the next phase, which is cytokinesis. Cytokinesis is where the cytoplasm has been equally divided. the organelles are being redispersed, and some of those are being kind of rebuilt. You begin to see the nucleus beginning to reappear because it's starting to be assembled. You can still see the chromosomes a little bit, but they're starting to unravel. And then when that happens, then we have what we call a cell plate formed. And that cell plate is in cytokinesis is kind of right here. So we're getting this new cell membrane being developed, and that would be cytokinesis. So let's look at the entire process in its entirety as we'll play it. Remember, it's a cycle, so it just keeps going from place to place. So we can really start just about anywhere. So we're going to start an interphase and just watch it go. It takes just a few hours to get it done. And then once it's divided, then everything can restart again. Remember, interphase is about 98% of the time. Prophase, you begin to see the chromosomes, or in particular the chromatids. In prometaphase, the spindle fibers are being attached. The nucleus is pretty much gone at this particular point in time. The chromosomes or chromatids are beginning to line up in the center. Along the center plate, we have chromosomes completely lined up. Spindle fibers attached to the centromeres. And as it begins to pull apart with the spindle fibers contracting, that's anaphase. And then in telophase, the chromosomes or chromatids have reached the end point on the poles. That's telophase. And in cytokinesis, when we begin to see the cell plate form, cytoplasm is equal divided. Nucleus is beginning to form around both groups of chromatids or chromosomes. So really that's it. If you really want to see this and have an opportunity to kind of work through the animation yourself, go to cellsalive.com. It's a great website not only for mitosis, but also all the things in the cell cycle, everything that has to do with the cell. So I hope that helps, and we'll see you later. Thanks. Bye.